us from. Good morning, good afternoon, good evening, wherever you're joining us from across the world. We are very happy to have you with us for this monthly academic pharmacy section webinars. And this is the July webinar holding in August, pardon us. And you're very welcome to join us for this session where we're looking at empowering young academic pharmacists and researchers with the use of statistics. You're very welcome. In today's webinar, Professor Arinola Joda is serving as the moderator, a professor from the University of Lagos, an EXCO member of the Academic Pharmacy section, and Vice President and Treasurer of the African Pharmaceutical Federation of the FIP. And joining me is, as co-moderator, is Dr. Jonathan Jato, a pharmacist and lecturer at the School of Pharmacy, University of Health and Allied Sciences in Ho, Ghana. He is currently vocational training coordinator at the School of Pharmacy, and his research interests include natural product drug discovery, antimicrobial stewardship, and neglected tropical diseases. He actively contributes to FIP Academic Pharmacy section and is actually the co-lead for the webinar sub-working group of the Research and Scholarship Working Group. So, but today we're here to be looking at this topic on introduction and overview of statistical and data analysis, empowering early career academics and researchers. Before I introduce our panelists, let me take some announcements. As usual, the webinar is being recorded and live streamed on YouTube, and the recording will be available along with other events that I've held on FIP on the FIP events website, www.events.fip.org. We have a question and answer box. We prefer that you put your questions for the webinar in the Q&A box, while you can introduce yourself and tell us where you're coming from in the chat box. You're welcome to provide any feedback on webinars to webinars@fip.org. And if you're not yet a member, you're very welcome to join us and become a member on the by linking with the FIP membership registration site on the FIP website. Again, just to remind us that there's a new translation feature on Zoom that FIP Congress Office is subscribed to. So if you want to enjoy that, you turn on captions, you enable the translation toggle, and then you choose your preferred language under my caption language so that you can follow along with the live translated captions. And we would like for you to tell us also in the chat box how you're enjoying the use of that feature so that we can document the usefulness of it thereafter. In today's program, we will be having the introductions, then we'll go straight to the presentations. Our first speaker will be taking us through introduction to an overview of statistical data analysis. Our second speaker will be telling us about choosing the right statistical test, something that many academics and researchers are still confused with. Despite many years. So introducing our first speaker now. She's Associate Professor, Founder, and CEO. She has a PhD, MSc, and a BPharm. Was officially recognized by the Pharmaceutical Society of Ghana in August 2005 for her pioneering role in clinical pharmacy education and practice in Ghana. Pharmacists to be appointed by the WHO Ghana, and the first to serve on the Ghana Health Service Council overseeing the public health sector. She was in full-time academia at the Touro University College of Pharmacy in NYC from 2008 to June 2025. She was elected chair of the Global Education Special Interest Group of the American Association of Colleges of Pharmacy for the 2021-2022 academic year and received the Outstanding Service Award in April 2024. She's an academic editor for PLOS ONE, editorial board member of the American Journal of Pharmaceutical Education, and a peer reviewer for 26 global professional and scientific journals, including the Bulletin of the World Health Organization. Currently, she's the founder and CEO of Crafters Global Communications, LLC. Join me as I welcome Dr. Addo-Atuah to take her presentation. Over to you, ma'am.
Thank you very much, and for the opportunity to give an overview of statistical analysis in research. But before then, we need to look at the overview. My topic is to look at the overview of research in general, and why it's important in the responsibilities of the academic pharmacist. We all know that in all schools, we are reviewed on three concepts or points, like teaching, scholarship, and service. Teaching, we all understand what teaching is, and scholarship will explain what it is, and service to the school, service to students, and service to the professional organization that we belong to, and service to the community. The next slide, please. So what comes under scholarship? Scholarship can be, looking at it, writing for grants to fund research studies, or grants to do projects, innovative projects in teaching or in the community or in practice. And then we come to research itself, and then publications, publishing your papers in peer-reviewed journals, and then editing manuscripts that have been submitted to peer-reviewed journals, and then being a peer-reviewer. All these things are considered to come under the scope of scholarship. And the importance is that it helps the academic pharmacists to progress, in their career path. And depending on the type of institution, they will have they weigh the three options differently. Some will weigh more in research, more in the scholarship, more in teaching and service. It depends on the type of organization that we are dealing with. Next slide, please. So in pharmacy research types, we have many, because we have many arms of the pharmacy profession. So we have the basic sciences research, also called the bench research, which we do in the lab. And then we have the practice or clinic-based research, looking at different, testing different health systems or healthcare delivery systems, looking at patients, review and all those things. And then in the population side, where I am, we have different types of research, like pharmacoepidemiology, looking at use of medications in the population and their outcomes. That's pharmacoepidemiology, using epidemiological principles. And then we have the pharmacovigilance, looking also in population, monitoring the use and adverse effects and whatever in the community. Then we have pharmacoeconomics, where you are looking at the cost and outcomes, comparing different options, different medications or different treatment regimens, and coming out with the most cost-effective to use in clinical practice or whatever. So what are some of the challenges that early career pharmacists, academic pharmacists may face? They may face inadequate exposure to research methods and data analysis, especially in the PharmD, that's where it's so much clinically focused. They may not have enough time to look at research methodology and all those things. And therefore, there may be uncertainty about choice of statistical tests, and difficulty in interpreting test results to make meaningful conclusions and inferences, and then balancing statistical test or significance with ethical and equity considerations in the population-based studies. For example, you may find that using a certain medication is only cost-effective in the elderly, but is it ethical, or is it ethical to leave them out because it's not cost-effective? That's what we are talking about here. So why is statistical analysis matters? Why is it important? By analyzing the data that you will collect, we can transform the raw data into evidence so that we can, based on it, to really make concrete decisions or policy decisions, or even practice changes and things like that. We can also use the statistical test to test hypotheses and support the clinical or scientific claims that we would have made even before, we started the research. So the statistical analysis will help us to confirm what we thought, or we thought was, the answer or whatever. So the test will give us the assurance that indeed this is true or not true. And then it minimizes the bias and enhances the integrity of the research. And then, of course, it also, once we give in the significance of the results, we can facilitate evidence-based practice. That is why we said evidence-based, guidelines for this, because they've done the research and they've shown that it really is true. And of course, it gives the foundation for impactful research. Next slide, please. So the factors to consider before starting the research. What are the research questions? What questions are we really undertaking this research to answer? And then what study design will be most appropriate and feasible to answer these questions? So you have to frame the questions first, and then the questions will relate to the type of study design that will be appropriate to answer those questions. And then what types of data need to be collected to answer the questions? So the type of data will also depend on the study design and things like that. And then what data collection methods or tools will be needed? And then what resources are accessible to the researcher? Things may be available, but not accessible. We can look at these resources as the time constraint. What time is available to the researcher? What population is available? Because it's not easy to just go out and start asking questions and whatever. So all these things come into play. What is the level of expertise of the researcher? All these are resources that are important. So in terms of study design types, we have quantitative design types, we have qualitative, we have mixed methods where we have partly quantitative and partly qualitative. And then retrospective, that is when we go back to in time to collect data to answer the research question. Prospective is when we are collecting the data as the research unfolds. And then we have what is called cross-sectional, that we are taking data at one time, at a point in time, that's the cross-sectional data collection. And then longitudinal, that is after the initial listing, it goes further, like a cohort study. You follow the population for some time. So depending on the type of question, the questions we want to answer, it will determine the type of study that we will, the design that we'll take. For example, my PhD research was looking at factors affecting the access to antiretroviral therapy in Ghana. And I used the qualitative methods. But inside there were quantitative as well. Qualitative, why? Because I wanted to see and experience the perspectives of different people who are connected with the antiretroviral therapy program. So having in-depth interviews with providers who are giving their taking care of the patients, and then the perspectives of the patients themselves. What do they, how do they experience the accessibility like? What distances they have to travel before they reach this thing? How do they feel, in terms of the interrelation between them and the provider? So this lent itself to qualitative, data collection and methods. And the methods leads, the method you choose also determines the data collection method. I mean, the design you choose will determine the data collection methods and tools that you will use. So qualitative normally we use, either focus groups, where if you want to look at a group of people with, either different characteristics of the same, or in-depth interview with key informants, interviews. So quantitative is where we have more of the, we are looking at surveys and things like that. So the difference is that in quantitative, you look at the breadth. I mean, you can ask questions from thousands of people. But then in qualitative, there's a selection of these thousands so that you get in-depth of their perspective, their lived experiences with the disease like HIV, for example. So that is where the qualitative is so rich. My dissertation, I have more than 500 pages. So you can imagine, it's a very big, because you gather a lot of information. And the mixed method is when you combine the qualitative and the quantitative. Then data types can be categorical or nominal. Then ordinal, like when you're asking, responses like, agree or, strongly agree, that's the ordinal. Then there's discrete or countable variables, and then continuous, where it can be measured over time. Next slide, please. So these are some data collection methods. I mean, this is not, I just gave some examples. If you look at the quantitative, normally, the surveys are the most frequently used. The surveys can be self-administered. Surveys by the participants themselves can answer the questions, or the researcher can administer the surveys, especially in places where we have people who may not be able to read or something like that. And then we can also have telephone surveys, which, depending on the resources available. And then we have the electronic data capture, using Qualtrics and things like that. In terms of the qualitative studies, the major ones are the focus group that I've talked about already, that we are looking at a small group of people to get their insights or perspectives on specific topics. And then one-on-one in-depth interviews, either with a structured, interview guide or semi-structured, Next slide, please. So other things to consider in research is, of course, you can't just get up and go and start asking people questions or things like that. You need ethical approval, and the institutional review board, is what most institutions have. And before you present your proposal for their review and and approval for you to undertake the study, of course, you have to write everything that we're going to do, the protocol you are going to use, the data collection methods and tools, how you are going to analyze the data and things like that. All these things will come into the IRB proposal that you are submitting. And then, of course, the informed consent procedure is very important. How are you going to ensure that the people you are going to talk to, I'm talking about people because I'm used to population-based studies. How do you ensure that they understand what you are asking them to do. How are you going to ensure their privacy and things like that? That all comes into the informed consent. How are you going to document the consent? Is it going to be, is this something that they are going to sign, or is it going to something that they will say, give the consent verbally? All these things will have to come into the proposal that you are sending to the IRB. But it's very, very important, the informed consent procedures. And then the type of population available, the sample size, all these things are things to be considered. The participant recruitment, how are you going to recruit? In fact, again, in that my research that I'm talking about, the ART, I had to use the counselors there, to recruit for me at the ART site, because you don't know the people. So there was a protocol. All these things were part of the IRB proposal that I sent. So everything was documented. I was going to get the participants and all that. And then protection of human subject in research is also something that is legally, required. In fact, there are laws, protecting human subjects. Laws where you cannot use children, pregnant women, things like that. These are all there, and you need to make sure that we follow the protocols and procedures that are, and policies and guidelines that are, there, which are, globally accepted, and specifically in your organization. So I just want to use an example of a research study with my students here in New York. We wanted to examine the accessibility of essential medicines in New York City. And New York City has got 5 boroughs. We have the Bronx, the Brooklyn, Manhattan, Queens, and Staten Island. So there were 2 target medications. Each were selected for asthma, diabetes, hypertension, and depression. And you ask, how did we come about it? Because these are on the top 150 list of medications that are frequently used in New York City. So they are. It means they are very much in use, and also tells us about the prevalence of these diseases in the U.S. population, and I believe it's in all other populations. Next slide, please. So what were the research questions? These were the research questions. Are there significant differences? We wanted to see whether there was significant differences in the mean drug prices across neighborhood income levels and boroughs in New York City. We also wanted to see whether the drug prices in a zip code, whether it's associated with the number of pharmacies in the zip code, or what, because we know something called a drug desert, where there are places where you can, travel 10 miles or whatever without seeing any pharmacy. So if these, isolated pharmacies, their medications are more expensive than where you get more pharmacies, we wanted to see whether there's any difference between the drug prices depending on the number of pharmacies in that zip code. And then the third question was, are mean drug prices in a zip code associated with the proportion? In US here, we have the independent pharmacies, and then we have the chain pharmacies, like CVS, Walmart too is there, and Duane Reade and all that. So we wanted to see whether these chains, the medications prices in the chains are different, whether they are cheaper or more expensive than those in the independently owned pharmacies. And then the other one was, are the difference between the drug prices in the independent? That's it. That's what I meant. Okay. So how did we analyze? Of course, we pulled the mean drug prices for, first of all, because we had available the different different neighborhoods, the zip codes, and the available information about these zip codes, we could categorize the zip codes into high, medium, and low income neighborhoods. And these neighborhoods also come with their associated demographics. I mean, there's racial group proportions, the percentage of people with insurance, the percentage unemployed. So you have an idea of the population you are dealing with. So we pull the mean drug prices for the high, medium, and low income neighborhoods to get a citywide mean drug price for each of the income levels. And then we can compare these above means for statistical significance across the levels using ANOVA, the one-way analysis of variance, because ANOVA, we are dealing with more than two groups. So that's why we didn't use t-test, but used ANOVA. And then we compare the mean drug prices across the 5 New York City boroughs, also using ANOVA. And then we examine the relationship between the number of pharmacies in the neighborhood and the mean drug prices using the Pearson product moment correlation. So whether there was any correlation between these. And then in terms of relationship between the proportion of independent unchained pharmacies in the neighborhood and the mean drug prices, we use a chi-square test. So depending on whatever the questions you are asking in a research, you may use different statistical methods in the same research because you want to answer the different questions. So that's what I wanted to let you understand, that you may not use just one research statistics to in a research. You may use, normally you have to use many to answer the research questions that you post before you starting the question, the research. So in summarizing, there are many factors that need to be taken into consideration before starting a research study. This is to ensure that the correct data collection method is used, the data type is used, and the appropriate statistical texts are used to arrive at the correct conclusion, to determine, to be able to truthfully answer the research questions. These are very, very, very important, to get your research questions down. And then you look at the research design, the data collection methods that you will use, sometimes quantitative, sometimes qualitative, sometimes mixed, depending on this thing, whether it's cross-sectional, longitudinal. All this depends on the type of research or the type of questions you want to answer. Mm-hmm. Okay. Next, please.
I think that's the last slide.
Okay. Thank you so much. Mm-hmm.
Thank you very much. Thank you so much, Dr. Addo-Atuah. I'm very sorry, and I have to apologize because some of the slides, the headings were just not there, and they were there before I maximized the slides. So I don't know. I really don't know what happened. But
Yeah.
Thank you very much. You were able to speak very well to your presentation. So we have taken the first topic and looking at that overview. And then we have our next speaker. Thank you very much, Dr. Addo-Atuah. We have our second speaker, Dr. Mahfoudh Abdulghani. He will be talking to us about choosing the right statistical test for research. He is an associate professor of pharmacology and he holds a PhD from the University SAINS, Malaysia. His research focuses on natural product pharmacology, toxicology, and reproductive health, with an emphasis on Uricoma longifolia and hormone-free contraceptives. He's an active member of FIP, AcPS, and CSPT, and he has authored over 40 peer-reviewed publications, achieving an H-index of 18 and contributing extensively to international collaborations in drug discovery and medical education. So please join me in welcoming Dr. Abdulghani for his presentation. And while he comes on stage, let's remember to put our questions in the Q&A box so that at the end of all the presentations, we'll be taking the questions from there. Dr. Abdulghani, you have the floor.
Okay, thank you for this opportunity to be with you. It's really very interesting, and we hope that we can have many. So the topics for my session is about choose the statistical test. And we try to focus, about three main points. The choice of statistical test depends on three main points: research questions, type of data, and then the data set, how many we have. For if we are supposed to be, we're already conducting the research and we have the data. So based on these 3 points that play a major role in identifying and selection and choice that statistical test. Please, next slide. So if we look for the first point, the nature of the research questions being investigated, that we can look for this question, what type of measuring or what the relation. So we can say, if we look for that, for the relationship, that do we have the true independent variable? So we have two ways will be, we have, if yes or if no. If yes, that means we need to measure the regression analysis. So I'm come from background of lab-based. If you want to do the regression analysis, most of it that we can measure normally we use this most commonly to make a regression equation so that we can measure the unknown sample after we do the standard substance for this analysis. If no, that means we need to measure the correlation, how to say, analysis, so that we can identify the relation between two variables, okay? So that can, identify this type of data. And then, and also we can see the differences. Sometimes the test for equal variance, if you have how many group we can be treatment, that we need to see the differences. As well as also in the research, we can make a comparison or comparing. If you have, for example, 2 drugs, we need to test, for example, for treatment of hypertension, or that can, so that we can see how is significant difference, so that we make this comparison. So based on this now, the research question is a very important from the beginning. When you start do the analysis, identify what type of approach you're going to take for this analysis. Okay, could next please slide. Now, if you look for the second point, the type and the scale of data being analyzed, I do this diagram so that can be easy for us to identify, as the previous speaker that mentioned about it. We have categorical, that means qualitative data, or numerical quantitative data on the right slide. So if you look from categorical data, either nominal or ordinal. If you say nominal, that means, for example, like, type of blood, so that we have, that we can say nominal. Ordinal, that when we see have, when measure the pain severity, mild, moderate, severe, this is ordinal. This data that we do, either we take chi-square or Fisher's exact test, that we need to, how to say, we need at the end from that to look for, to analyze this categorical data, specifically to examine the differences between observed and expected frequencies, for example. And also the Fisher exact test is also a statistical significant test used to analyze the relationship between two categorical variables, particularly when dealing with the small sample size. When we look for the other part or the other type of data, numerical data, we have two types of data, discrete or continuous. Discrete data, that means countable variable, like for example, number of visits, you can say, is not a continuous. But if you look for the continuous measurable value, like when you say a high temperature, so that we can frequency measure, okay? And in this quantitative data, we have two types of data can be generated. And this is very important, that can identify what type of test that you need to use. And that you have, should your data either normally distributed or non-normally distributed. If the data is normal distributed, then we can go for t-test if two group or ANOVA. If the data type of data is non-normally distributed, so that we can go for non-parametric like Van-Whitney and U-test or Kruskal-Wallis. It depends on the number of group. So now we can see from this type of data, they can give you a beginning of type of general test that you need to go, or the approach of statistical test can use. Okay, next slide, please. So now if you look for the third point, choosing the right test now. Now we previously, when we do during our master or PhD, we look for type of research that have similar, then we see what type of statistical test used, but without, we don't have this strong in statistical background that we need to identify why we choose this one. So if we know that our data is parametric test, so that we need to identify, is our data is independent group, we bring this data, or is extracted from independent group or from paired group. This is another point. So that if independent group, so that you have to see how many group we have, either two group or more than two group. If two groups, then we can go for independent sample t-test. If more than two groups, then we have to go for one-way ANOVA, and then we have to see if there is any significant difference, because one-way ANOVA, they give you the general. If there is any significance between any group, but they cannot identify which group, so that we need to go for, after that, if there is any significant, then we can go for post hoc. And this is one of the, mistake that can be done. You do ANOVA, then do post hoc, even though there is no significant difference between group. So this we can say many papers they do. But this the one way ANOVA, they identify that you can go for next step or not. Because after one way ANOVA, you have to select the post hoc test, and post hoc test will be depend on other factors also. Okay, so if our parametric data from paired group, so that if two group, we have to take that test, and if our group will be more than two groups, then we can go for repeated measure ANOVA. And this idea, they can give us how to say the proper way to identify. Someone, they tell me, okay, what the difference between independent simple t-test or pair test? If I use the same, we can see maybe we can run wrongly, but we can see sometimes is the data, the sensitivity data, and also the outcome maybe can be wrong conclusion. Okay, please next slide. So now, if our data is non-parametric data, so what we need to go? Also, the other factor is the number of group. If two group, then the equivalent, the t-test in the parametric test and parametric data, I can use Van-Whitney for non-parametric data for two groups, if only we have two groups. Okay, here. But Wilkinson sign for ranking test, because normally for non-parametric, we do ranking data. If we have more than two groups, then I can go for Kruskal-Wallis test, because only they can, see the more than two groups, I can go for Kruskal-Wallis t-test, so that can give you this sample. This is the how we know we choose the suitable test that for non-parametric test. Or someone they tell me, okay, if I don't want to use sometime, parametric test, they not prefer because, they not give us significant, they would like to have using parametric test. Yes, can be. Should we need to convert it our data from nominal to numbers so that we can, how to say, or logic, so that can be normal distributed, and we can go for a parametric test. Okay, next slide, please. Now we can see from this, when we compare between parametric and non-parametric used for squeeze and rank data, for parametric example for t-test and ANOVA and Pearson, non-parametric example for Manwitni, you are critical wireless Spearman. This is the example of t-test. For parametric test, we have to go for one-way ANOVA. If our group, it depends on your study and experimental design or study design. If you have a one control and you have several groups, you need to compare to the one control, then we can go for post-hoc, then it test. If you want to have a multi-comparison, so that maybe we can go for another test. And this two factors very important. The homogeneity test is very important to select either which type of post-hoc test that can go for parametric test. Okay, next slide, please.
Can you not see the slide? I've changed.
Okay, so based on that, we can see how to say, satisfaction score between three clinicians then we can use for critical wireless. For example, if you would use for correlation or personal Spearman, then we, for example, age versus cholesterol level. Okay. Next, please. Yeah, so in conclusion and summary, we can see how to say that choosing the right statistical test either can select the test based on research question, data type, scale, and number of groups. Very important. Data type, either categorical versus numerical. For parametric test, we have t-test, and we know t-test can be two types, depend on the data generated from dependent or bed group. ANOVA and Pearson need the normal continuous data. For non-parametric test, so that we can use Mann-Whitney, Pascal Wallace. Okay, and this is for ranking data. This end, thank you very much for your listening. I think I kept to the end.
Thank you very much, Dr. Abdulghani, for your clear presentation on choosing the right statistical test. I believe your slides really spoke to the topic, and we are grateful for your presentation. As we have informed before, all questions will be taken at the end of all the presentations. So thank you, Dr. Abdulghani. We move on to the next topic, and we're moving slightly away from generic statistical testing, and we're moving on to systematic reviews for evidence-based decision-making. And we have Dr. Mahfoudh Loganathan, an associate fellow at the Institute for Big Data Analytics and Artificial Intelligence. He'll be talking. She's an associate member of the Royal Pharmaceutical Society of Great Britain and an active member of the academic pharmacy section. She's a recipient of a National Ministry of Higher Education grant for the project titled Risk Stratification Modeling of Inappropriate Non-Steroidal Anti-Inflammatory Drug Use for Osteoarthritis using Predictive Deep Learning and Algorithm Validation in high-risk community dwelling older adults. She successfully supervised PhD projects. The most recent graduated with thesis entitled Development, Testing, and Application of a Medication Adherence Tool for Intervention Assessment in a Geriatric Population. Please join me as we welcome Dr. Loganathan to give her presentation. Over to you, ma'am.
Thank you very much for the kind introduction, Prof. Arinola. Hello, everyone. Hope you're all doing well. Actually, could I have the first slide, please? So the one just before this.
This is the first slide.
Okay, no worries. So I have a subtopic. It actually says practical steps to conducting and analyzing systematic reviews and meta-analysis. So I will go through some of these practical steps, and hopefully by the end of my talk, you will be able to understand how systematic review and meta-analysis are important in evidence-based decision-making. So for just a second, evidence-based practice often relies on two key research methods, systematic reviews and meta-analysis. Both aim to summarize existing research, but they do so in distinct ways. So systematic review is a structured process where researchers collect and evaluate all relevant studies that meet predefined criteria. The purpose is to answer a clear research question in a comprehensive and unbiased way. Meta-analysis is a statistical technique that combines numerical data from multiple studies. The aim is to produce more precise estimates of the effect and identify patterns or differences across studies. The processes involve taking eligible studies from the systematic review, extract the relevant quantitative data, and apply statistical models to pool results. The outcome that you can expect is a statistical summary, often shown as forest plots, and this will enhance conclusions of individual studies. Next, please. There are essentially 5 key steps when reviewing literature effectively. Step 1 is framing the research question for the review and protocol development. You will often hear terms like PICO, inclusion and exclusion criteria, and the AI tool that can assist you at this stage is Gemini. Next, step 2 is to locate the potentially relevant studies and perform screening and then selection of the studies to be included in the review. There are a few AI tools that can help you at this stage, such as PubMed GPT and Rayon AI or Covidence. They can assist you with the screening processes. Step 3 is to assess the quality of the literature, and one will often hear phrases like risk of bias assessment. ROBVIS, ROBVIS, is one example of an AI tool that will prove helpful at this stage. Step 4 is to summarize the evidence. Here, the studies are grouped by themes, interventions, or outcomes, and the results can be presented qualitatively in charts or tables, or quantitatively, where a meta-analysis is performed. For statistical synthesis, programs like R, Jamovi, and Stata will be handy. Next, please, Prof. A high-quality systematic review begins with a clear, focused, and answerable research question. One of the most widely used tools for developing such questions is the PICO framework. P denotes population, the group of individuals of interest. I, intervention, the treatment or exposure being investigated. C, comparison, which is the control group, placebo, or standard treatment. O denotes outcome, the specific effect or result which is being investigated. Next, please. By applying the PICO framework, researchers ensure their question is specific, relevant, and methodologically sound, which is essential for guiding the literature search and subsequent evidence. To give you an example, population could be adults with gout. Could you please hit play? Thank you. To give you an example, population could be adults with gout. The intervention could be febusoxstat, and that could be compared to another urate-lowering agent, say allopurinol. And the outcomes of interest could be, say, cardiovascular deaths. Therefore, the research question could read, does febusoxstat increase the risk of cardiovascular mortality when compared to allopurinol in adults with gout? Next, please. Thank you. Once the research question is established, a comprehensive search of two major biomedical databases is performed, PubMed and Scopus, in this demonstration. Search parameters could have some limitations or restrictions to them. For example, the English language only, if you could please hit play, thank you. Search parameters could have some limitations or restrictions to them. So for example, the search could be restricted to only English. You may or may not apply any date restriction to the date of publication of the articles. The study design could be focused only on RCTs, given their high level of evidence. So therefore, you would exclude case reports, narrative or systematic reviews, editorials, conference abstracts, and non-English articles. The search strategy is developed to capture all relevant studies corresponding to each of the PICO elements. So for example, gout could be termed hyperuricemia in one study, and another study could call it high urate level. Usually Boolean operators, for example, and or are used to combine the terms to maximize the chances of retrieving the relevant studies. Next, literature screening is conducted using Rayon AI. This is a web-based platform that is designed for systematic reviews. Here you can see that the project can be shared between two independent reviewers. So this allows blinding, independent decision-making by individual reviewers, and therefore bias is reduced. Before screening begins, duplicate records that were retrieved from PubMed and Scopus are removed using Ryan's duplicate detection feature. This step is taken to reduce redundancy. You can see the total records here. What you see right now is the process of removing duplicates. Rayon AI is, it can be automated, but it still requires human intervention at the end. So it requires you to mark as to whether you agree to its decision. Okay, here the reviewer is selecting whether to include or exclude, and there is also an option to have it undecided as for now. Okay, the records after duplicates are 323. The remaining 323 records are screened by title and abstract by two independent reviewers. Rayon AI will flag articles which had conflicting decisions. So if one reviewer said yes and the other marked as no, Rayon will flag those articles and the reviewers will then discuss and decide on a final outcome. So the records after duplicates that remain are 323. These are screened by title and abstract by the two independent reviewers, after which the reasons for exclusion is also recorded. Okay, the diagram that you see on screen is a Prisma 2020 flow diagram, documents the number of records that remain after exclusion at each stage. And the reasons for exclusion are also documented to ensure transparency and reproducibility. So some of the reasons cited here are wrong study design, wrong publication type, wrong population, foreign language, and wrong outcome. Okay, so after full text screening and further retrieval of articles, usually through a reference list, the final number of studies to be included in the review is 6. Next, please. Thank you. The revised Cochrane Risk of Bias tool for randomized trials, ROB2, assesses 5 key domains of potential bias with judgments characterized as low risk, some concerns, or high risk. Domain 1 looks at bias that arise from the randomization process. Domain 2 looks into bias that is due to deviations from intended interventions, also known as performance bias. Domain 3 looks at bias from missing outcome data, or also known as attrition bias. Domain 4 looks at bias in measurement of the outcome, detection bias. And domain 5, bias in selection of the reporting results, or selective reporting. To go into each detail, let us have a look at the next screen.
Did I hit the play button?
Sorry, is it finished? Maybe if you could just move the cursor to the midpoint. Yeah, okay, that would do. Thank you. So to go into each assessment, so for example, the assessment of, say, random sequence generation, allocation concealment. So this means that the doctor who's in charge should not, or the researcher should not know which patients belong in either placebo or intervention group. In the CARES trial, allocation was randomized and concealed from both researchers and participants, and baseline characteristics were similar. Therefore, the judgment is low. The risk of bias assessment for adherence to intervention, blinding of participants and caregivers, and the application of intention to treat principle. So in the CARES trial, you can see it was double-blinded and participants were analyzed in their assigned groups regardless of treatment completion. Discontinuation rates were balanced between febuxostat and allopurinol arms and were unrelated to unblinding. Therefore, the judgment is low risk of bias. As for the assessment of completeness of outcome data and handling of missing data, in the case of CARES, it reported high loss of follow-up, 45% in the febuxostat group and 39% in the allopurinol group. The possibility of unrecorded cardiovascular deaths after discontinuation raised some concerns. Therefore, the judgment is high risk of bias for cardiovascular mortality outcomes. As for the assessment of objectivity of outcome measures and blinding of outcome assessors, in this case, an independent adjudication committee blinded to the treatment allocation consistently assessed cardiovascular deaths. The judgment is therefore low risk of bias. The assessment for selective reporting of outcomes and post hoc changes in outcome emphasis. In this example, cardiovascular death was specified as a secondary outcome but received heightened focus after results. Therefore, this indicates possible harm. And the judgment is some concerns. Following that, because the Excel tool stores only one study at a time, data were compiled and uploaded into an online ROB2 traffic light plot generator to visualize judgments across studies. As a summary, most studies were rated as low risk or having some concerns. With the exception, White 2018 unfortunately had a high overall risk of bias driven primarily by high attrition bias, domain 3. From this visualization plot, the traffic light plot clearly displays the domain level judgments for all included studies, aiding the transparent appraisal of the evidence base. Next, please. Thank you. And now I would just go through the key individual trial findings. Firstly, for McKenzie et al., published in 2020, febuxostat was non-inferior to allopurinol for composite CV outcomes. No increase in cardiovascular or all-cause mortality was observed. In the CARES trial by authors White et al. In 2018, non-inferior for composite cardiovascular endpoint, but showed higher all-cause mortality with a hazard ratio of 1.34, and this significantly increased cardiovascular mortality 1.22 with febuxostat. In the FAST trial, few cardiovascular death events were reported, not a primary endpoint, no clear difference detected. In the CKD Feather by Ordell et al. 2020, no significant difference in serious adverse events, suggesting comparable cardiovascular safety in CKD in patients with CKD. As for Suzuki et al. In 2021, febuxostat reduced oxidative stress markers, but showed a numerical increase in hospitalization for heart failure. Finally, for Baker et al., published in 2010, trend was favoring febuxostat. This was likely due to inclusion of younger patients, hence they are categorized as patients with lower risk. In general, all three of them had showed that there were no significant associations with cardiovascular events, except for two of the articles. I will go into details in the next few slides. Okay, review manager, I will call it RefMan from this point onwards, is used to pull study results. The effect measure is denoted as odds ratio, OR, with 95% confidence interval. The statistical model that was used is Mantel-Hansel method under a random effect model. The rationale for using this model is that it is suitable for rare outcomes and accounts for inter-study variability. Here, data entry is being made. The extracted number of cardiovascular deaths and total participants per arm from each study is imputed. From here, number of studies are 6 RCTs. The pooled odds ratio is 1.16 with a 95% confidence interval with a 95% confidence interval ranging from 0.92 to 1.47. The p-value, however, is 0.2, indicating that it is statistically not significant. The heterogeneity, I denoted by I square, is 13%, indicating low heterogeneity, and this shows that there is good consistency across the studies. The interpretation of this forest plot is that there is no statistically significant difference in cardiovascular death risk between febuxostat and allopurinol. Okay, next, please. Okay, just to summarize very quickly, we analyzed data from 6 major RCTs comparing febuxostat and allopurinol, focusing on cardiovascular death as the primary outcome. Together, these studies enrolled 16,550 patients, predominantly male. Prof, if you could just pause this screen for a minute, please. I think I might take a bit longer. Okay. Together, these studies in total comprise 16,550 patients, predominantly male, and they were aged between 52 to 71 years. Participants were mostly from Western countries, with one Asian population included. Comorbidities such as hypertension, diabetes, and chronic kidney disease were common, reflecting a high cardiovascular risk population, representative of real-world gout patients. Okay, yes, if you could hit play, please. Okay, from this forest plot, you can see that only 2 studies, Baker as well as Odell in 2022, favored febuxostat. Okay, the overall odds ratio is 1.16 with a fairly narrow confidence of 0.92 to 1.47. This indicates that patients on febuxostat had 60% higher odds of cardiovascular death compared to allopurinol. However, the confidence interval crosses the line of no effect, which is denoted by number 1. This indicates that the result is statistically not significant.
Thank you so much.
Okay.
Thank you very much, Dr. Loganathan. That's your last slide.
If I can just conclude.
A very interesting presentation and presentation style as well.
Yeah,
So,
Dr.
please,
Prof,
Jen.
if
Yes,
I could
please.
just conclude on the last point there. So what we want to say, if you could just hit the cursor on the last, at the last second, please. Okay, from this review, we can see that the febuxostat may be a reasonable option for patients who have allergy to allopurinol. However, we do not have conclusive results to suggest that it is entirely safe. It just means that this review just signifies that we do not have conclusive evidence to suggest that it is associated with cardiovascular deaths. Thank you.
Thank you so very much, Dr. Loganathan. I've learned many things in this presentation today, starting from the use of the AI tools in carrying out the systematic reviews. Thank you so very much. And I can see our audience are very well engaged on the 3 presentations that we have had so far. We have a couple of questions in the Q&A box, and we'll be moving on from here to the questions and answer segment. So can I invite all our panelists and the co-moderator to put on their videos and let's have a good discussion. We have about 25 minutes, thereabouts 20-25 minutes to take on like 10 questions that we have on the Q&A box. So I'm going to stop sharing so that we can have everybody on screen for this Q&A segment. Thank you, everybody. Okay, interestingly, Dr. Loganathan has mentioned some of these, some tools that are relevant for systematic reviews. And I'm going to be asking Dr. Jato to tell us of some other tools that are generally useful for statistical analysis and in research. Over to you.
Okay, so thank you very much. As we have listened.
Dr. Jato, can you hear me?
Sorry, just a sec. Can you hear me? There was another device close by which was giving a feedback.
Yes,
so
can, but
if you can hear me, as we listen to the presentations, one key thing we are taking home is the fact that before you start research, you must have key interest in what the outcome is, and that determines what research tools to use. in terms of the research question to set, in terms of the type of data collection tools to use. But more importantly, when you collect the data, analysis is key, because collecting the data is just half the work. Data analysis becomes the step that converts the data into evidence, into information. And so depending on the data collection tool, the kind of data you have, the kind of research questions you have, there are several tools that we can use in processing our data to make meaning. Some of them are general. For example, we have what we call the SPSS, which is used for descriptive statistics, t-test, ANOVA. You can do regression and non-parametric tests. We also have Stata, for example. Stata is a good tool for epidemiological studies, for biostatistical evaluations or assessment. We have what we call the SAS, which is used especially for the pharmaceutical industries when they are processing large data sets to be presented to regulatory bodies. It's very good for processing large data sets. Then we have some from the open space, which are free, that you may not have to buy, that you can use. One common example, as mentioned, was R. You can use R, which is a coding software. And one advantage of R is that we have a large community of people using R. Also, when you hit a hurdle, you just put it on the community of users, ask questions, and people who have used it have evidence, and they're able to support you. The other one, for example, is what we call Jamovi, and I think Loganatha mentioned this. Jamovi is very effective. It's used as an alternative to SPSS, and sometimes when you use Jamovi, you can integrate it with R. So you start with R, you have issues, or you want to verify the data, you can go into Jamovi, and you don't have to start afresh. We also have JASP. JASP is used for Bayesian analysis. It's also a free software that is available. You can just go online and get. And just like Jamovi and also for R, you have a lot of people using it. So when you have challenges, they are available to assist you. Then you can come to the regular spreadsheets where we use Excel or Google Sheets for basic analysis. If you want to just have basic observations and you are not interested in doing deep analysis, you can go with the regular Excel or Google Sheet. For qualitative data analysis, there are some keys. For example, we have this one, MaxDA. MaxDA is very effective for qualitative studies. You may have to register for it, but I know they have also a free option that you can get, just that you can analyze all kinds of data, but they have a limit. They also have Atlas TI or Atlas.ti, which is effective for qualitative data analysis. Then we have special domains, which I have often used, is the GraphPad Prism. GraphPad is very effective if you want to have nice images. When you finish processing and you want to have a very beautiful presentation of your figures or your tables, your graphs, then GraphPad Prism is the place to go. Again, we have various variations of it. Some are paid for that you may not be able to get depending on the level of analysis you have. We could also use EpiInfo, for example, when you are gathering data on the field and you want to do both qualitative and quantitative analysis, you can use EpiInfo. I've mentioned also the PSPP, which is a variation of SPSS, but that one is quite difficult to use in my perspective. Okay, in my perspective, that is quite difficult to use compared to the R, which is a coding software, and there are a host of others. One key thing I have to leave here is that your choice of the data analysis tool depends on your study design, your study questions, and also your convenience, because these things you have to learn by yourself. So there are some that are more complicated to use, some are easy to use, so you start somewhere. I started using Stata, but I had challenges. I went to use R. In fact, I've learned a lot of them, and depending on the data I have, if it's from the lab work, the tool to use is different from when we do some field work. And I think anybody who is starting or interested in processing data should start by learning the software. Just go online. There are tutorials. You can also do short courses on, for example, Coursera and EDX and other short courses on this. There are short courses for R, short courses for Stata, for SPSS. So depending on the one you choose, you do a short course and you get you improve your skill and become best at it. Thank you very much.
Thank you so very much, Dr. Jato. Jonathan.
Arinola, would you please stop your slide sharing so we can see your faces? Thank you.
Okay, I thought I did. Oh, sorry, I thought I stopped sharing. Sorry, I thought I stopped sharing since. All right. Okay, thank you, everybody. And thank you, Jonathan, for that very extensive list of tools. And this is just to tell everybody that depending on what you want to do, there are a lot of help. There's a lot of help that you can get. Some of them free, many of them free actually, and some of them that you need to subscribe to. And then you may even find that your institution is actually subscribed to some of them. And by linking with your university library or your institutional library, you may have access to some. The key thing, as Jonathan has said, is that you have confidence in using these tools because you know data. If you put something in, you will get something out, whether it makes sense or not. Something will come out and you may not be able to defend it, but you will certainly get something. Okay, so before we look into the Q&A box, and please, any of our panelists, you can please go to the Q&A box and be responding online because I have a couple of questions I want to ask before we go there. For Joyce, I wanted to ask you, you talked about IRB, ethical, can you hear me? Hello? Can we hear me?
We can hear you.
Yes.
Okay. So you talked about getting the ethical considerations, getting IRB approval. And I wanted to ask for the benefit of our participants, is ethical approval also required for animal studies? I know that that one is an obvious yes. The one that I am not very sure, and I think there will be some information on, is when we're doing laboratory studies that we're not using animals, we're not using human beings, do we also need to get ethical approval for that? So from the animal studies and then into the laboratory studies. Thank you.
Yes. The only, in fact, even studies that don't require active participation of human beings, you actually submit to IRB and the IRB will give you the exemption, like using, data, already established data, collecting it to analyze, you still submit it. I mean, that's what we do, but then they will say this is exempt. So they give you that exemption. So especially, I'm a reviewer, so I know that all the peer-reviewed journals, they always want you to see whether the paper submitted has any indication that they were submitted for ethical consideration. So that is very, very important. So whatever type of study you need to submit, then the IRB will determine whether this is exempt. So that one you can get the exempt, within a day or even half a day or something. And then depending on the extent of human involvement, they may actually, have the full review, and give you the approval. So it depends. But then I would say that whatever study you are doing, especially if you want to publish, send it to IRB and they will give you the exemption or the full approval. It's very important.
Thank you. Thank you very much. The next question goes to Dr. Abdulghani. You talked about determining some of the steps that one will do based on if the data is normally distributed or not. So could you give some explanation as to how one will check for normal distribution?
Okay.
Before one can apply the test?
First of all, for the normal distribution, if the data or the sample size is quite high, we can consider the data is already normal distributed if the sample size is very high. But sometimes, especially in the experimental studies, the number, how to say, for example, the number of animals, if you want to measure for any response or for the drug or something, it will be due to the ethically, you cannot use so much of animal, and also impossible to do a big group, because sometimes you use 4 or 5 group per experimental design. So if you want to measure the normal distribution, then there is a test through normally I have experience with using SPSS, so that I can measure the normal distribution of the data. But and also normally, if you previously, when theoretically, should your data have a bell shape, so that your data can be, focus on the main, can be almost in the middle, that we can measure it through the SPSS for normal distribution. If that data is a normal distributed, then okay, you can go for both. The important of normal distribution, that can prevent the bias of the significant, because sometimes we are not aware. And even though sometimes maybe we can conduct the, how to say, ANOVA test regardless for the result, either normal distributed or not, if the data is the same for non-parametric test and parametric test. But theoretically is not, because non-parametric tests only they can measure the ranking. But for parametric test, you can measure the differences here. I hope that I can answer the questions. Thank you.
Thank you very much for that. Mathumala, I have a question for you as well. In carrying out systematic reviews, is it necessary? I'm not sure the word is necessary. Is it compulsory that one uses a guide? I know you mentioned using Prisma in as part of your presentation, but is it compulsory that one uses a guide that would capture all the various aspects of this of the review, or just focusing on the PICO, using PICO, and then responding based on that would suffice?
Thank you for the question. Obviously, there's a lot of a depth of information when it comes to systematic review and meta-analysis. So one part is performing the review and analysis accurately. The second part is actually writing it up. So when you write a manuscript, actually, let's move one step back. Protocol should be developed initially. So this is if you're targeting high impact journals, you want to have it published in good journals, and you want to have to reach a wide audience, you should ensure that your protocol is firstly published. So databases that accept the publication of protocols quite simply and easily would be like Prospero. Okay, so they have their own guidelines as to how the protocols should be written. As for the review and meta-analysis, Prisma 2020, I believe, is the latest guidance. In fact, right now there is an AI tool, Prisma Checklist AI tool, that will assist to ensure that you have met all the criteria, the checklist. So starting from the abstract, all the information which are required from stating whether two reviewers perform the search, as well as the how was the write-up done as well. So all these information are required by the Prisma guidelines to be included from abstract up until the end of the manuscript. So I think it is highly preferable, and it would be good to follow this guidance.
Thank you. Thank you very much. Trying to go to the chat box, there are quite a number of questions. A few of them have been answered. There's a question about handling missing data for categorical data. And okay, I can't add that to any question. Is anybody, Jonathan, would you like to answer that question? How do we handle missing data for categorical data?
I'm not particularly an expert in this area, but I have experiences. So it depends on the level of the missingness. So for example, when you have categorical data that you, in the process, mix some variables or some data specifics that will not affect your analysis, you can still go ahead. I've had an example where we collected data, but when we came back, we saw that some data that we were supposed to have collected with the tool was missing. So we looked at the research question and we're still able to process the data without failing to answer the question. Yes, but I'm not really an expert in this area and I don't know much about how to handle this. But I've had an experience before. Yeah.
All right, thank you. I don't know if anybody else has anything to add to that before we move on to the next question. Okay, Mahfoudh is answering the question on excluded data. And you have answered the question on Likert scale. There's a question about McNeema test. I don't know if anybody knows that. I've personally not heard about it before. It says, when can the McNeema test be used in place of chi-square test? Does anybody have experience with McNeema? Nah. Okay.
I haven't come across it.
I've not come across it either. Okay, there's a question by Mola. In the era of AI, how the statistical data analyzing softwares like SPSS can still be used for analyzing? I didn't get this question. It's best that I put this question, should please edit the question. I'm not sure what the person is asking. How the statistical data analyzing softwares like SPSS can still be used for analyzing pharmaceutical data? We can still use
I'm sure
SPSS.
the person wants to know whether.
Yeah.
I think the person wants to know whether AI has can replace these tools. If that is the question, then
yes. But you must know what you're doing with it.
Yeah, most of the companies even are adapting to AI, so the tools themselves are becoming enhanced with artificial intelligence so that it can improve the processing power and the analysis that you get. And so AI does not directly replace it, but rather augment these existing tools.
All right, thank you. I'm smiling at the last question I'm looking at, and we have 3 minutes to end the call. This attendee says, as a pharmacy student, which statistical skills or concepts is the most primordial to master? I was talking to somebody, an MSc student yesterday, and I say relatively many of us are not very strong with statistics. A lot of us learned by fire, maybe during our PhDs and other postgraduate programs. And for some of us that developed particular interest, we then went further to really master some of it. So when this student is asking which is the most primordial of the skills to master, I can't say which, but what I can speak to that is that whichever of the tools that you decide that you want to use, I think learning about the various aspects and the various offerings of that tool will help you to do a better job for the next time that you need to analyze data. You may start off with just doing, of course, the descriptive analysis is basic. You know your mean, you know your mode, you know you've done your standard deviations and standard error of means, as the case may be, and then you're then moving it up. Okay, you may take just one question from your project to try and find out to try and carry out some inferential analysis just to determine if there is a statistical difference or an association Once you have been able to get that done, you then layer on that, add more to that, moving on to things like correlation, things like regression, and so you do it in layers. You can't learn everything at once, and you won't start by trying to know the toughest bits before the basics, So you take it one layer at a time. You start with descriptive analysis, you move on to inferential, the basic inferential statistics, chi-square, t-test, and then you move on with that, depending on how sophisticated the research questions that you then want to answer continue to be. So I'll leave this question, I'll pass this question around in 1 minute. Let's each talk to that topic before we end the Joyce, I'll start with you.
Yeah, I think as I alluded to it, we need to, be more intentional in introducing at least the research concepts into, the PharmD, for example. We've tend to be so much more clinical, leaving this aspect. But if you don't understand the basics of research methodology, what is when something is statistically significant, the p-value, whatever, all these things should be introduced and as part of the the PharmD, the basic pharmacy course. That as you move forward, to do your residency or whatever, or your master's or PhD, these things will come. But then I think that we should start introducing basic research knowledge into the pharmacy course. Is very, very important. Otherwise, how do you understand somebody has written a cost-effective paper? I mean, how do you understand what they are talking about?
What they're talking about. Yeah, I would say that, I can speak for Nigeria. At the undergraduate level, we have introduced research methods and research methodologies as part of the program. But I think, just as you've said, we need to be more intentional about giving that course more practical and applied significance with our students. So by the time they're in final year, we expect them to do some of these things, and the basis that they have is not very strong, but it's there. It's in the BPharm program, it's in the PharmD program here in Nigeria. And I believe if it's not in all programs, it really should be, because the relevance and the importance of your projects, your studies, your research work can only come out when you can analyze properly. Mathilama, 1 minute on this topic before we close.
Nothing really to add.
Okay,
I agree.
all right. Thank you. Abdulghani, you have something to add? Last thoughts and last words.
Yeah, I agree. But when you look from point of view of education, the limited time for the student that overloaded with especially for
pharmacy.
So, and also when you look for the statistics, is about application applied. But I agree with that should teach the student the applied concept in statistic rather than to give the theory, which they just knowing but not applying. So that you need, a lot of, brainstorming so that we can come up with some courses, or maybe like short workshop for the student out of the class, because rather than, doing in the class, I feel that is only very minimal student they can get. Thank you.